We cannot just do the class silent. So if you people speak, I will be silent in that time. So that will help me. So some active participation will be uh, uh, very much appreciated. Okay. Since Iram is also a little under the weather. How are you, Iram? How are you doing?
Yeah, I'm better now, but I do have throat infection.
You also have a throat problem. This is the time. Yes, Devangi and Waziha. I think you both have to hold the fort along with me today. Okay. And so about that, let's begin with a quick summary. And who would like to give a quick summary among Devangi and Waziha? Devangi,
Yes, sir.
would you like to give a quick summary, please?
Yes, sir.
Yeah, thank you.
So, ATP and NADPH are basically used inside the plant cell, and the first stable product.
But the question is where did they came from? Where did ATP and NADPH came from?
From the product of photosystem.
Yeah, the light reaction.
Yes, sir.
Yeah. Okay. Right. So the
And
question is where it is used. So to solve that, we have to understand the biosynthetic pathway. Yes, continue.
so the biosynthetic pathway is also known as Calvin cycle, as it was discovered by the scientist Melvin Calvin. And so basically, he identified that the first stable product in the photosynthetic pathway was PGA, or 3-phosphoglycerate.
Mm-hmm.
But some plants had another first stable product of carbon dioxide fixation, that was oxaloate.
What is the
And that
difference
is a,
between the two?
yes, sir. So the PGA is a 3-carbon organic acid, whereas oxaloacetate is 4-carbon.
Mm. Okay.
So the plants that follow the 3-carbon pathway are called C3 plants, and the other type are C4 plants.
Mm-hmm.
But the Calvin cycle takes place in both C3 and C4 pathways.
Yes. Okay.
Then the primary acceptor molecule of carbon dioxide was found to be a 5-carbon molecule, that is RUBP, ribulose biphosphate. It is basically a 5-carbon ketose sugar.
Mm-hmm.
And the enzyme or the protein that helps to catalyze this carboxylation is rubisco.
Mm-hmm.
That is the most abundant protein found in the biosphere, and its full form is ribulose biphosphate carboxylase oxygenase.
Yes. It has dual capabilities, both to carboxylase the RUBP and also to oxygenase the RUBP. But we will be studying in Calvin cycle, we will be studying about the carboxylase activity of it. Okay.
Yes, sir. So Calvin cycle consists of 3 major stages. It is the dark reaction. So it is carboxylation, reduction, and regeneration.
Carboxylation.
Yes, carboxylation.
Yeah.
So first of all, the inputs that we have, carbon dioxide and water. So from that, carbon dioxide is accepted by RUBP, and After carboxylation, it forms 3-PGA.
Okay.
And the enzyme is Rubisco for this. Then this 3-PGA, 12 molecules of 3-PGA are formed, and then they are
If you
reduced
take 6
into,
molecule of CO2, that you forgot to mention.
yes sir, 6 molecules of CO2 and 6 of RUBP. Then reduction takes place, in which we use the ATP and NADPH, 12 each, and they are converted into ADP, inorganic phosphate, and NADP plus. And after reduction, the product is glyceraldehyde 3-phosphate.
Yes, it is a triose phosphate because it has 3 carbon. So we have not changed the number of carbon. If you see, 3-phosphoglycerate also has 3 carbon, and triose phosphate also has 3 carbon, but we have reduced the molecule. We have added more hydrogen to it.
Yes sir, so 12 G3P is formed, and out of which the 2 will leave for the formation of glucose, and rest 10 will undergo the regeneration process. So
Regeneration of what?
of RUBP.
Yes.
So but before that, an intermediate is also formed, that is ribose 5-phosphate, and the enzyme that helps is
RUBP
phosphokinase.
has how many carbons, Angi?
5.
Okay. And glyceraldehyde 3-phosphate has how many carbons?
3.
3. And how many molecules of RUBP we are regenerating back?
6.
So how many carbons do we need? 6 into 5, 30. And do we have 30 carbons when 10 molecules of glyceraldehyde 3-phosphate did
Yes sir.
not leave and continued? Yes. Now do you understand that why 10 continued and only 2 are allowed to leave?
Yes sir.
We have to make a balance between regenerating RUBP and also to make glucose, right?
Yes sir. And to form ribose 5-phosphate, also 6 ATP are converted into 6 ADP, and then the regeneration occurs, and finally we again get 6 molecules of RUBP.
So here we are just balancing the equation. We are putting 6 and 6, but now we just know that when it happens with one CO2 at a time, right?
Yes sir.
So basically, how many cycles are required to make one molecule of glucose?
6 turns in the Calvin cycle.
6 turns. Okay.
That
And how many
means, yes, 18 ATPs and 12 NADPH
are used,
Yes.
utilized? Very good, perfect. And today we have to begin with the C4 pathway, correct?
Sir, one more thing you told, that the first NADPH molecule is committed to reduce 1,3-biphosphoglycerate.
Yes, correct. And the first ATP is, I also told you that at any given step in a biological, biosynthetic, or any a biochemical pathway, if we use ATP, that means what? We are making the step irreversible, correct?
Yes, sir.
So the steps which require ATP commitment are irreversible. And of course, it should be irreversible. It should make sense. If you have spent energy, you better want that work done, right? You don't want it to just come around incomplete or undone.
Yes, sir.
Correct? Perfect. So now let's begin with see if I also ask you to go back and read about Cran's anatomy, bundle sheath cells, mesophyll cells. Did you do that?
Yes, sir.
Okay, so please help me and lead this discussion today. So let's talk about the C4 pathway today. And as you told, the C4 pathway is present in the plants that are adapted for dry conditions. Okay, plants which are in dry tropical environments, write down C4 pathway is present in plants which have evolved and adapted to dry tropical conditions. Okay. Another thing is, in these plants, as Devangi also mentioned, the first stable product the first stable CO2 fixation product is, what is it?
Oxaloacetic acid.
Oxaloacetate. So I will write OAA, oxaloacetic acid, which is a 4-carbon molecule, correct?
Yes, sir.
But and the third thing is that though they have oxaloacetic acid as the first CO2 fixation product, they do use C3 pathway, or which is known as Calvin cycle, as their major or main biosynthetic pathway. So the point is, the main biosynthetic pathway of even C4 plants is Calvin cycle only. So which means Calvin cycle, which we just mentioned above, takes place in both the plants. The only difference is that when you look for the first stable product of CO2 fixation, in C3 plants it's G3P, and in C4 plants it's PGA, and in C4 plants it's OAA. PGA, right? Yes, phosphoglycerate. And in C4 plants it's OAA, oxaloacetic acid. But at the core, both of them use Calvin cycle. So what is the difference? How are C4 plants any special, better or inferior than C3 plants? How are they different? What is the speciality? What's special about C4 plants? What's special then, people? Those who went back and read about the Cran's anatomy and bundle sheath cells can tell better.
Sir,
Yep. Yes, Wazir.
C4 plants lack a process called photorespiration.
Before going to that, C4 plant, but how do you know that they lack? Just because you figured it out after reactions?
And
But
they
what is the
tolerate high temperature.
okay? But what's special? What's special in terms of the plant?
They have a special type of leaf anatomy.
Yes,
Anatomy.
correct. Now you are hitting the right note. So what do we mean by having a special type of leaf anatomy? What special type of leaf anatomy you are talking about? Just give me 1 second. I'm just putting my charger in. Yeah, you continue. Tell me, what is this special type of leaf anatomy you are talking about? What do they have? Do they have something that C3 plants don't have? Apart from the abilities to do this and that, structurally, is there something? Yes,
Uh, so
yes,
they
Seema.
have wreath-like arrangement.
Sorry, they have
Wreath-like arrangement.
what like arrangement? Sorry, I could not. What arrangement?
Wreaths in
Wreath.
crans
Okay.
anatomy.
Yes. Yeah, you mean crans itself means wreath. It means the wreath-like anatomy. Okay, so they have. But what is this wreath-like anatomy? The term is not clear itself. So clearly, the special thing is. So sorry. The special thing is that they have a special. Just one second. They have a special leaf anatomy. Now, what is special about this leaf anatomy? So what they have is they have something called bundle sheath cells. Okay. Now, do you understand what is a sheath? Sheath is a layer. Uh, so they have two types of cell. On the top, they have a. I should not say top or bottom, but they have one cell which is called a mesophyll cell. And then they have another cell. It's called the bundle sheath cell. Is it clear? And something which I think Vaziha or Devangi also mentioned about these plants are they can tolerate high temperatures better. Also, what else did you say? They show response to high intensity lights because where they live in that region in the dry tropical conditions in the tropical region, sunlight is available for a very long time in the day, not like the um, temperate or the tundra region where the sunlight is very less in the tundra region near the Arctic and Antarctic. In the tropical region, the sunlight is very much available. So they show a response to high light intensity. Okay. One more thing is that someone said that they lack photorespiration. Correct? Anyone knows what is photorespiration? have not studied this yet, but anyone has an idea what is photorespiration? No idea what is photorespiration?
So
Yes, tell me.
So in that case, I'm not sure, but Rubisco starts capturing oxygen instead of carbon dioxide.
Rubisco starts to capture
Oxygen instead of carbon dioxide.
oxygen instead of carbon dioxide. For what? To what? What was it capturing carbon dioxide for in the Kelvin? First step of the Kelvin pathway. You are correct, Iram. You are on the right path. So why was it capturing carbon dioxide? 5. So the enzyme Rubisco, what was it doing? It was fixing the CO2, right? So instead, if she's right, if oxygen is used, or if plant starts to utilize oxygen instead of, it is just like respiration, but it is happening in the presence of light. You may not be knowing photorespiration, but you must be knowing what is respiration in your own cells. Can you tell me what is respiration? Respiration, people. Can anyone tell me what is respiration? It is a process of utilizing oxygen to break down glucose. Okay, or you can say that the process of breaking down glucose to make energy in the presence of oxygen. In other words, you can say oxidative breakdown of glucose in the cell to produce energy. Correct? So we are using oxygen in the process of respiration. Plants do photosynthesis in which they produce oxygen, but sometimes they also do photorespiration in which instead of like they start utilizing oxygen. So Rubisco, in this case, the PGA binds instead of CO2, the first stable product, it binds with O2 to form one molecule of phosphoglycerate and phosphoglycolate. Okay, phosphoglycolate is a 2-carbon molecule because instead of CO2 we are using O2, so one C is missing. Okay, so we cannot if one CO is missing, we cannot make another 3-carbon molecule. So one is phosphoglycolate, and this pathway is called photorespiration. So in this pathway, we do not produce glucose, we also do not produce ATP, but we release CO2, just like a normal respiration that human does. Plants also do this. This is a waste kind of respiration where CO2 is released, but in our respiration we are at least producing ATPs. We are producing energy. Here there is no ATP synthesis, no NADPH synthesis. Instead of CO2 we start using O2, and instead of O2 release we plants start releasing CO2. But do you think it's, what is the benefit? What is the biological function of this kind of pathway? Why are plants doing photorespiration? What is the use? Any idea? We don't know. It's still under research. We don't have a clear answer. We have some we have some answers which may or may not be true depending on what we find that the plant biologists. I don't know where the field currently has reached in terms of this research. For sure, there are some insights, but C4 plants completely lack photorespiration and they do better in dry conditions because it is a waste of energy. It looks like that C3 plants, which can show photorespiration sometimes, it is because Rubisco has both functions of acting as a carboxylase, and it can also act as a oxygenase. So sometimes it starts acting as a oxygenase rather than acting as a carboxylase. So when it does that, you utilize O2, release CO2, but you do not end up making ATP or NADPH. So it does not make sense why a plant is doing that. But C4 plants have evolved better and they have completely stopped doing that and they are doing good. So people also think that it's just a glitch in the system, you know, and C4 plants took out that bug glitch. So it's a better, it's some advanced, or what you can say, updated version of C3 plants. It can be true. It might be true also. Okay.
Sir,
Yes, Devangi.
sir, so what is broken down?
What is broken down? Nothing is broken down. We don't broke. If we were just, it's just like we cannot complete the Kelvin pathway. Okay.
Okay, sir.
It's not like our respiration where we take oxygen, oxidize glucose, release energy, store that energy, and do our work. Because that is a useful thing, no? If we are doing any catabolic activity and producing energy, that's a useful thing. Here, we are losing one carbon, getting oxygen, because of which we are not able to make two molecules of phosphoglycerate. So one molecule we have to compromise with a phosphoglycolate. And that's why the pathway cannot complete, because we will never be able to make 12 molecules of this glyceraldehyde 3-phosphate, out of which 2 we can use for glucose making and 10 we can send back for regeneration. So this all will just stop. Do you understand? That's photorespiration. We will study about photorespiration in detail later on. Is it clear till now?
Yes, sir.
Now the type of but let's focus for now on this leaf anatomy. Now write down, C4 plants have a special leaf anatomy where there are large cells around the vascular bundles in a C4 plant. There are large cells around the vascular bundles in C4 plants, which are known as bundle sheath cells. Bundle sheath cells. So this is happening in the leaf. Okay. These bundle sheath cells, this one, which I'm talking about here. Sorry. These bundle sheath cells forms many layers around the vascular bundles. What are vascular bundles? Can you tell me, people?
Xylem and phloem.
Yes, water vascular bundles. Okay. Yes. So if you are sheath means covering. So multiple layers of these bundle sheath cells are present around the vascular bundle. Okay. And these bundle sheath cells have one thing. So they have a very large number of chloroplast. So first they have large number of chloroplasts. Second, these bundle sheath cells seems to be like, it's like taping around the duct. So if you have in your houses, if you have a sink pipe or something, and now you start taping around it, many layers of a tape. So it's, you can understand in what conditions do we do that? When would you like to tape around a tube or a pipe or a garden hose which you use to water your plants? When you want to prevent any leakage, right?
Yes, sir.
So, and makes sense, right? Because remember, where are these plants doing very good? Where are they situated? In dry tropical conditions. In dry tropical conditions, what is one thing which is precious than gold?
Water.
Water. So plant would like to not waste water, and everything should be used. So to make sure around these vascular bundles are these big multiple layers of these bundle sheath cells. That's why they are called bundle sheath. They are present around the vascular bundle. So first characteristic is they have large number of chloroplast, and second characteristic is that they are impervious to any exchange, even gaseous exchange. They do not exchange anything, which means they have no intercellular space. What do we mean by intercellular? Word inter and intra.
Between the cells.
Yes, so between the cells normally there is space, right? Cells are not very tightly packed together. There are spaces between the cells. Things can diffuse between these spaces, but in this case there is no intercellular space. Okay. Now we understood what is bundle sheath cell, correct? Now what is the benefit of having a bundle sheath cells? Is what is this? What is the benefit? So remember, there are mesophyll cells as well, correct? Now these mesophyll cells are the cells which actually receive CO2 from the atmosphere. So atmospheric CO2 is received by the mesophyll cells. Then from the mesophyll cell, it goes into the bundle sheath cells. But I told you, there is no intercellular space. How does the CO2 go then? If there is no space between the cells, what is the other way through which cells can still, exchange things, transport things among themselves? There was one structure that we studied about. In the chapter cell, I told you.
Plasmodesmata.
Perfect. There's something called plasmodesmata. Plasmodesmata becomes even more important where the cells have a cell wall. Correct? Plant cells have a cell wall, which is a rigid, proper, rigid physical wall. It acts like a wall, okay, a solid. And between the cells which have cell wall, plasmodesmata can become a very easy tunnel-like thing through which things can be exchanged. So that happens. The CO2 enters into the bundle sheath cells through small pores known as plasmodesmata. Is it clear, everyone?
Yes, sir.
Now, what are the characteristics of the bundle sheath cells? That these have large number of chloroplasts. Now, if they have chloroplasts, they will be having chlorophyll. So what they can do? They can carry the photosynthetic pathway, the biosynthetic pathway as well, correct? So this is what happens. So let me make a better diagram. So this pathway, which includes the Calvin pathway in itself, is called as Hatch and Slack pathway. It was figured out by Hatch and Slack. What is it called? So for C4 plants, we study something called Hatch and Slack pathway. Now what happens in Hatch and Slack pathways? There are two types of cells in C4 plants. I'm making cells as very can I insert shapes? Okay, no. Oh yes, I can insert it. So this is one. Okay, and I'm using the line color as dark green. I'm using blue. Insert. Okay, people, can you see this?
Yes, sir.
Let's say this is the mesophyll cell. Can anyone who has studied tell me what is mesophyll cell? This is the yes. Anyone can tell me what is a mesophyll cell? What is mesophyll? What is mesophyll? No, so you did not studied. Okay, so the homework stands. You have to go back and figure out what is a mesophyll cell. Why we call it mesophyll? These are simple, very basic things. If you do not find, then come to me and then I will tell you. So what happens is the CO2, I should say the atmospheric CO2, enters into the mesophyll cells. And when it enters into the mesophyll cell, the first molecule, which is so what was the first in case of this C4? What was the first stable product of CO2 fixation?
Oxaloacetate.
Yes. Now, oxaloacetate is a C4 or 4 carbon. So, what do you think is the first? Should be the first acceptor? Acceptor molecule should be how many carbons? In the above case, in the Kelvin cycle, the acceptor molecule was a 5-carbon molecule called RUBP. But it was 5 because it was accepting one CO2, and then from one CO2 there were 2 PGA formed. From 6, 12 PGA were formed as the stable product, correct? 5 plus 1, 6. The maths work there. Here we have to make a 4-carbon molecule as a stable product. 1 carbon is coming from CO2. When this carbon comes, okay, it is converted into carbonic acid, HCO3. Still it has only one CO, HCO3 negative. And this is received by a 3-carbon molecule called phosphoenol pyruvate, and all this is happening in this mesophyll cell. Phosphoenol pyruvate plus HCO3. So this is a 3-carbon molecule. Total how many carbons are there now? HCO3 plus
4.
phosphoenol pyruvate, 4. So this is the step where fixation happens. And as a fixation, we get OAA, which is a 4-carbon molecule. Okay. This is called C4 acid. Oxaloacetate is a C4 acid. Is it clear, everyone? 1 plus 3 total, oxal 4. Is it clear? The first step is fixation. Now, through some pores which are called plasmodesmata. So I'm showing in red here the plasmodesmata. And this is all. Through this OA is transferred into bundle sheath cells. So now this C4 acid comes here. Is it clear? So can you understand why are we doing this? Can you tell me what is the benefit of doing this? We do in the first place. Okay, let me complete it first. Then we will discuss about things. So this C4 enters here. And the interesting thing that happens now, so this phase through this was called transportation or transport. This is the second step. First you have to fix it, then just transport it into the bundle sheath cells. In the bundle sheath cells, what will happen is decarboxylation. Now this is very puzzling, but in biology everything must have a reason. If it is benefiting the organism, everything must have a reason, which is to benefit the organism. If it is evolved like that, most of the things are like that, except for photorespiration, which we have not able to understand till now. But here what happens, the third thing is decarboxylation. Now in decarboxylation, what we mean is that this C4 acid goes, so OAA goes, and from this CO2 is removed back, and what is left? If you remove CO2 from COA acid, what will be left? a C3 acid will be left, right? Yes or no? Now this C3 acid will be transported back through the plasmodesmata into the mesophyll cell again. And now you know which is that C3 acid. What is it? It is regenerated into this phosphoenol pyruvate, which is again a 3-carbon acid. Phosphoenol pyruvate means we can also call it phosphoenol pyruvic acid, right? PGA, phosphoglycerate or phosphoglyceric acid, oxaloacetate, oxaloacetic acid. So this phosphoenol pyruvate is the same 3-carbon acid which is regenerated. Make sense, people? Do you understand this whole thing?
Yes, sir.
Now, the thing is, my question is, what did we do here? It's very confusing. You took CO2, we thought you will fix it. Yes, you did. We fix it into a 4-carbon acid. Okay, cool. Maybe you are not happy with the 3-carbon acid. Then you transported it as a C4 acid into the bundle sheath cell. Still cool. But then you just do the reverse. You remove the CO2 which you fixed with a 3-carbon molecule. You remove the CO2. You made that C3 acid again and transported it back so that it can do the same thing again. Which means everything which is being done to obtain this CO2, right? Because this CO2 will now go into what? It will go into the fixation by Calvin cycle. So Calvin cycle is happening here. Where? In the bundle sheath cells of the C4 plants. And you know what is Calvin cycle? This is Calvin cycle. So CO2 now enters and RUBP is present here in the bundle sheath cells. So everything that it's a normal Calvin cycle. So here it behaves like a C3, right? Now look carefully. I give you 2 minutes. If any one of you figure out why did the plant went through all of this funny business, you understood the whole photosynthesis then. What do you think is the benefit of this? Ultimately, the plant did the same thing, fixing the CO2 by Calvin cycle to make glucose. Then why did it fix it here? Why did it do this additional work? You fixed it here, made OA, transported OA, then was not happy with OA somehow, and then said, okay, it's like someone asked you to wear a blue shirt. You're like, no. You kept away the blue shirt. You started trying on all different kind of shirts and dresses. Then in the end, you're like, okay, chuck it. Let's wear the blue shirt. It seems like that, right? But it's not like that. What's happening here? Waziha, Devangi, Iram. Can you think? Can you think what's happening here? Very intelligently. This C4 plant, okay, which has this kind of anatomy. So this anatomy called Kranz anatomy, where this bundle sheath cells is surrounding the vascular bundle and the mesophyll cell. So very intelligently with this design, this C4 plant, remember in the Calvin cycle, who receives the CO2? Now keep giving me answers. Who receives the CO2 in Calvin cycle? Quick, people.
RUBP.
RUBP. And what is the enzyme that catalyzes this reaction?
Rubisco.
Rubisco. So Rubisco makes CO2 bind with RUBP, or in other words, I can say Rubisco makes Rubisco carboxylases the RUBP. Can I say that?
Yes, sir.
Now the problem with RUBP was it can also bind to oxygen and not CO2. And I just told you what happens when what happens when Rubisco prefers oxygen and not CO2. Instead of carboxylating RUBP, it will oxygenize RUBP, right? Because it's an oxygenase also. You understood? People, please, if you'll cooperate with me for the next 5-10 minutes, I won't be able to hold it for very long. So we will end the class after finishing this. So it has to be short. So please answer.
Sir, RUBP will combine to oxygen.
Yes. Rubisco will make oxygen bind to RUBP and not. So first the enzyme binds to the thing. Then it makes it bind with something. Enzyme does not participate in the reaction, it just facilitates. You must be knowing this, right?
Yes.
Enzymes are catalysts of biological reactions, biochemical reactions, sorry. In a pure chemistry lab, you use catalysts. So catalysts do not react themselves in the reaction. So Rubisco, instead of preferring CO2, if it prefers oxygen, the whole pathway will go waste because photorespiration will happen. Instead of making ATP, you will not make any ATP. You will also just not make any NADPH, no glucose, no sugar. So do you think it is useful for a plant to do something of this sort if it is living in a very testing conditions like a dry condition?
No, sir.
No, right? Yes. So what C4 plant decided is, see, we cannot trust Rubisco, okay? So let's do one thing. Let's make a different cell where we will keep Rubisco in isolation. Rubisco will not be allowed to interact with oxygen. We will not provide Rubisco with any oxygen. That's why bundle sheath cells are impervious to gaseous exchange, so that no oxygen can come in on its own. The only thing we will give the Rubisco is CO2. And how to make it sure? Do not let Rubisco receive the CO2 from the oxygen, because if Rubisco receives CO2 from the oxygen and give it to RUBP, or if RUBP receives CO2 from oxygen, it can also receive O2 from atmosphere, right? Because in
Yes,
mesophyll cells, things can just come from atmosphere. They can just diffuse in CO2, gaseous exchange can happen from the atmosphere. So CO2 can come in, oxygen can also come in. So they said, let's not allow this Calvin cycle happen in the mesophyll cell. Let's make a different specialized cell where we will control and ensure that only CO2 will meet Rubisco and RUBP, not O2. That way, none of the Rubisco will choose O2. None of our system will go into photorespiration. We will not waste energy. that way, or we will not undergo a futile pathway where we are not making ATP, not making NADPH, not making any sugar, but we are doing all sorts of things. So we don't want to do that. C3 plants do that. Let's be better than that, better than them, because we need to be better than them, because we are the plants of arid and dry conditions. So instead of RUBP, they made phosphoenol pyruvate, which is a 3-carbon molecule, receive the CO2 from atmosphere in the form of CO3, HCO3 negative bicarbonate, fixed it into oxaloacetic acid. That's why we got the first stable product as 4-carbon acid, because this was a false. It is a sham. It is a scam. It is just to show, or what to say, it is temporary. It is just to transport it into the bundle sheath cells. Once it comes into the bundle sheath cells, then remove the CO2 and provide the CO2 into the C3 cycle, and that this C3 acid can be transported back and regenerated and can do the same thing. So it's like you hired another man just for the purpose of picking CO2 and bringing it to you. So it's same thing like you went to a market to buy something and you could not differentiate between, let's say, one variety versus the other. You are both carboxylase and oxygenase, so you can sometime bring the right variety, sometime you can bring the wrong variety. So you said enough of doing this. Sometimes I bring the wrong variety, and then all the process is fruitless, useless. So I will make a person go to the market for me who knows this variety, will bring that variety only. I will just sit in my bundle sheath cell and get that variety without the fear of getting the other variety. Is it clear, everyone? Devangi, Vaziha, and Iram?
Yes, sir.
This is how C4 plants are better, or they can do better in arid conditions. Okay?
Yes, sir.
Yeah. And this is also how they never undergo photorespiration. So they lack photorespiration, because photorespiration is not a useful process for the plant as we understand it. So write down photorespiration. Let me end with photorespiration today. Is it clear to you, Iram and Vaziha?
Yes, sir.
Okay. So write down, in C3 plants, sometimes oxygen binds to rubisco. Oxygen binds to rubisco instead of CO2. Because rubisco has a dual function of being a carboxylase and also an oxygenase. Because rubisco has a dual function of being both a carboxylase and a oxygenase, so it can bind to both. But phosphoenol pyruvate can only bind to bicarbonate, which is carbon dioxide. It cannot bind to oxygen in this context. Okay. This decreases the CO2 fixation in C3 plants. This decreases the CO2 fixation in C3 plants. And instead of getting 2 molecules of PGA instead of getting 2 molecules of PGA, the C3 plant now gets one molecule of PGA and one phosphoglycolate, not phosphoglycerate. One molecule of PGA, which is a 3-carbon molecule, and one molecule of phosphoglycolate, which is a 2-carbon molecule, because RUBP had 5 carbons only. The 6th carbon it was getting from CO2, but if Rubisco, instead of getting CO2, gets O2, then you don't get that carbon. So now you have just 5, and you break 5 into 3 plus 2. So you get one PGA and the other phosphoglycolate, 2-carbon molecule. And that's why the Calvin cycle will not continue. You will never be able to make 12 3-phosphoglycerates now or phospho-3 glycerate now. So you cannot make glucose, and you cannot also regenerate RUBP. Is it clear, everyone?
Yes, sir.
Write down, this pathway is called photorespiration. This pathway is called photorespiration. So photorespiration only happens because of the oxygenase activity of Rubisco. So Rubisco's dual nature is responsible for photorespiration. And write down, in photorespiration there is no sugar synthesis, no ATP synthesis, no NADPH synthesis. So nothing is synthesized, but on the top there is expenditure. CO2 is released and ATP is used in that process. CO2 is released and ATP is used in that process. Okay, so it's a waste of energy for the plant. Okay, and last thing is, in C4 plants there is no photorespiration. Okay, and now you understand how. Because Rubisco's dual action is responsible for photorespiration, and we have locked Rubisco in this bundle sheath cell, which is impervious to any gaseous exchange. So only the gas that we bring to it, which is CO2, it is bound to bind with that only. Okay. So I think I won't be able to speak any further. People, I'm so sorry. So we'll have to stop here for today. Just 15 minutes earlier. I hope you all are okay with it.
Yes, sir.
Yeah. Okay. Thank you so much. And in the next class, we will be doing factors that affect photosynthesis. So we understood photosynthesis. We understood the light and the dark reaction. We understood both type of biosynthetic pathway, the Calvin and the C4. We realized that C4 is just slightly tweaked, but at the core, the biosynthetic pathway in both C3 and C4 plants are the same. We understood photorespiration and how it is avoided by the C4 plants as well. Please go back and revise all of this, and I'll see you all in the next class. Okay.